Thank you very much. When I got the phone call a couple of days ago to ask me if I would fill in for somebody who'd pulled out, it was obviously a great honor to be invited here as a second choice. And I was told it had to be a TED-type talk, and I honestly had never, ever seen a TED talk. So I went onto the internet, and I looked up TED, and I came to this film called TED. And I won't be referring to that further this afternoon. Reflecting on what both Liz and Nicola had to say about learning, reflection, and mentorship, some of you have heard this story before, but I'm taken back to my school days, and our mathematics teacher came into the classroom and said to us, he said, "You people, you don't know how stupid you are. You really don't. In fact, you're so stupid, I'm going to demonstrate it." And he challenged us. He said, "Stand up if you think you're stupid." And we all sat still, of course. We're not that daft. Until Michael, who was sitting on my left, slowly got to his feet. I was horrified. And the maths teacher said to him, "Michael, tell me, boy, you don't really think you're stupid, do you?" And he said, "No, sir, but I couldn't see you standing there on your own." So, and I recognize I'm the only one standing up at the moment, so I shall be brief. If I said to you, "Rare well done," rare well done, of course, refers to something we might think of food. And whilst you were all sleeping last night, somewhere in the order of 120,000 eggs were delivered into London, 2.25 tons of beef were delivered into London, and somewhere in the order of 250,000 chickens were delivered into London. And given that each of those, or so the thinking goes, could carry a pathogenic organism that might affect humans and also might bear bugs that are resistant to the drugs that we use, maybe we should be slightly worried. And certainly my mathematics teacher, which then developed into an interest for epidemiology, drilled into us that if you could count something, then you could say something about it. If you couldn't count it, then you were always going to be on a back foot. But what he didn't say, and what I think I would like to share with you this afternoon, is that it's the understanding the context, the structures of the populations, and the processes that are just as important, particularly when it comes to something as important as antimicrobial resistance. And there is perhaps nothing greater than the tsunami of articles in the popular press, and indeed in the scientific press, than perhaps the risks that are going to be coming our way if we can't find a solution to the issues of antimicrobial resistance in what will be a post-antibiotic era. And the disease that will affect us we will not have seen for several generations. And the one thing we don't have, of course, is time. But I'd like to turn the clock back a little bit. It was funny sitting listening to Ian, and I'm sorry he's not here. He had a picture of Ian McIntyre at the end of the left-hand side of sitting on the boat rail or whatever it was. And actually, when I started the work that I'm going to demonstrate now, back one of the papers that I read in 2006, I was actually sitting in what had been Ian McIntyre's office. So there is a circularity to today for me. But this paper that was published, as say, in 2006 from McMaster, demonstrated in a way that I hadn't really thought about before that these were researchers who'd taken isolates from the microbiome. They'd taken it from the soil, and they had then looked at isolates, and then they'd looked at resistance of those isolates to a range of antibiotics. And you can see that each of these black dots is one of the isolates around the circle here, and then these are the antimicrobials to which they're resistant. And you can see there's an enormous amount of resistance out there, but probably more importantly, that the diversity of that resistance is phenomenal. So we started by asking the question, well, if there's such diversity out there, what is it like if we look at humans and animals? And so basing a study on surveillance, and I acknowledge both Brian and Roger's comments about the importance of surveillance, based on the work of others, we looked at a huge range, over several thousand isolates that had been taken from beef cattle and from humans in Scotland during a 15-year period. And what we discovered was that not only was there widespread salmonella in those populations, in fact, it was for the epidemic of through the 90s and into the early 2000s, that there was salmonella to be found in both populations, but the diversity of that resistance in those salmonella were markedly different in the two populations. And there was a lot more diversity in the human isolates than there was in the animal isolates. Now, if you've got 25 different types of flowers in your garden, and I've got 3 in mine, the chances are you didn't get the other 22 from me. And that's the observation that we made, and that caused quite a stir in the scientific and, in fact, in the lay and medical communities. So what was it that was happening? Were these salmonella and their resistance determinants coming from the human population or the animal population, which was, of course, contrary to popular belief? So the other thing, besides DaCosta, who published his stuff in Science, you have to have a very pretty picture to get something into Science. Well, this is our pretty picture. I've told you about the phenotypic stuff, I've told you about the diversity. What I want to demonstrate now is that when we went to look at the genomic aspects of it, so we're now using whole genome sequencing, rather than the dots around the circle, if you imagine that for each animal and human isolate, we've now got a tip to this branch here, and this phenotypic tree illustrates the whole genome sequencing, if you like, identifying the hereditary, the life story of the evolution of those salmonella. And for each one of these, at each tip, you'll see one is blue, which is an animal isolate, and one is red, which is a human isolate. And where, when you look back through the tree, you can see it going from red to blue, or blue to red. So here we've got one that's going from red to blue. There are others which go from blue to red. That's where the salmonella has moved from one population to the other. And you'll see that there's evidence that the salmonella, not just the resistance determinants, but the salmonella are going in both directions. However, and this is the other important piece here, this figure here demonstrates the number of times that the salmonella has gone from one population to the other. And so we can see here the red and the black. These are where the salmonella are going from humans to animals or animals to humans. And you can see it's relatively rare. Number of branch-to-branch transitions very low. And up here, where it's remaining within the population, animal to animal or human to human, is much more prevalent. So on the back of the diversity work that we've done, we were also able to show that salmonella in general circulate within their own species, host species. And that although they do cross from animals to humans and humans to animals, it's a relatively rare event. And that also caused a bit of a stir. And we published that some 6 years ago now. So what did that tell us? And I am coming to the real message of the story for me. It told us that, yes, of course, antimicrobial use in animals is important, antimicrobial use in humans is important, but it's not as simple as the direction of flow being one from either animals to humans or humans to animals. The only way you can really explain what we observed is that there is an enormous amount, potentially environmental source, about which we know very little. And that really means that, of course, medics, vets, lab-based scientists are probably not going to provide the answers that we're looking for. And that the solutions lie in considering that animal-human environment. If you imagine each of these circles having animal-human environment, animal-human environment, animal-human environment in each of these layers, that we have to think about things in a much more inventive and stratified fashion. And the United Nations, the FAO, Food and Agriculture Organization of the United Nations, looks at it in 3 levels. They talk about the technical level, which to some extent I've just presented to you, the science. They talk about the social level. How do we change behaviors? Do we look at how we prescribe? As a profession, do we look at how we prescribe? I think there's been some remarkable advances in the reduction of usage in our food animal practitioners, quite remarkable in our farmers. And then finally, do we put our backing behind it? Do we get intergovernmental organizations, and do we get governments, and do we get the RCVS, and do we get all the regulators involved at the same time to make sure that when these 3 are considered together, that they come together in a way that is truly what I would call a One Health solution. Now, you have no idea how long it took me to put that slide together. So I'm going to do it one more time. People usually applaud at this point, by the way. And it is available for a small fee. So, but it's really, my take-home message from this is that the solution to antimicrobial resistance, and it's worth bearing in mind that today we've got people on strike because of climate change. There's another thing that's going to require a One Health approach if we're going to do something about it. It's going to require many disciplines, not just our own discipline, to come together. And so in closing, and I think we've heard about surveillance, we've heard about the comparative biomedical science, we had the question about public health, and we've learned about reflection and mentorship, and we had some 8 really remarkable talks this morning. And it's funny, but science tends to recognize individuals, and I know that each of us that have been humbled to be recognized today as fellows, it's important. But actually, it's not the receiving of the fellowship that's important. It's about becoming part of a fellowship, because it's only as a fellowship that we're going to make the impact that we know we can make. And like most things, like any sport that most of us play, team games, not only are they more fun, they tend to get the job done. Thank you very much.